We're going to chat about this table now. I kind of made a summary of microbiology milestones. Just to give an idea, it was really hard to do this table. I'm not complaining though, because it's sort of, I selected those things. I selected those discoveries and technical changes that happened over the years, but somebody else may find something else more fascinating or more important for the development of microbiology. So it's entirely subjective. It's my take on the milestones. But I wanted to make, we'll talk about them and we'll see why I think it is relevant. First of all, people knew about infectious diseases ages ago. For instance, in China, they practiced variolation. Smallpox. Have you heard about the disease? Smallpox? People get pustular rash, like little bubbles with a fluid in those bubbles in the skin. The Chinese used that fluid essentially kind of sort of as a vaccine. So they infected healthy people with that fluid, and it was a full-blown smallpox, but it was kind of less dangerous than acquiring it regular way, by a respiratory route. So people who underwent variolation had 1% chance of dying. Having a smallpox is 30% chance of dying. So variolation provided some additional benefits, okay? Of course, the Chinese had no idea that it was a virus, particles, or whatever. They just noticed that people who had smallpox and survived do not get it second time. That's where the variolation came from. Actually, Greeks, Romans, those guys, the Hippocrates in Greece and Varro in Rome, Hippocrates proposed that there are natural causes of the diseases. Not that gods are angry at you, okay, but something natural happened. And Varro actually suggested that there's some little things, minuscules, that can cause the disease. Again, they never had microscopes. They didn't know. And finally, Leeuwenhoek and Hoek, it was beginning of 17th century. Leeuwenhoek was the first to actually propose the microscope. He was a tradesman. He was working with glass. And Hoek first introduced the microscope as we know it now. The concept of the microscope as we know it now. And that allowed us to find out there are many creatures. And then in one of the milestones for the public health in microbiology was Edward Jenner in 1796 did first successful vaccination. He noticed that milkmaids who acquired the cowpox never get sick with an actual smallpox. So he collected some cowpox sample, like fluid from cows infected with cowpox, and inoculated I think he was a 9-year-old named James Phipps. And a couple of weeks later, inoculated the same boy with actual smallpox. Boy didn't get infection, he survived first vaccination. The name vaccination comes from Latin for cow, vacca. But you may ask, what's the difference between vaccination and variolation? Anyone? What is the difference between vaccination and variolation? Yes? It's not dead, but you're on the right track. It's weak, okay? It doesn't cause disease, like full-blown disease in humans, like very mild disease. So in vaccination, bless you, we never use the actual pathogen. Does that make sense? So Jenner was essentially the father of vaccinology. as Jon Snow, hello to all fans of Game of Thrones, was the father of epidemiology. In London, in something like beginning of 19th century, there were outbreaks of cholera. You know what the cholera is? You shitting like never in your life. Every 20 minutes. People can lose up to 20 liters of fluid a day. Yes. So they die of dehydration quickly. Other than that, a person can sit in the IV fluids for a week and the person is cured. But because you cannot substitute those 20 liters by drinking electrolyte. Especially they taste like crap. So Jon Snow was curious why there's a clustering of cholera cases in London. And he found out that a group of cases originated in people who used the same water well for the drinking water. And he made the conclusion that that water was contaminated with cholera. So he actually was the first to introduce that tracing of cases, trying to figure out the socioeconomic factors that contribute to the spread of the disease. Although all those developments didn't answer the main question, where do these microbes come from? Can life originate by itself? And for a long time, the theory of spontaneous generation was pretty common. You know, that joke that mice can arise in a pile of dirty socks. Well, that's pretty much something like that. So the idea was that living things can just pop up. And you see it is crossed here. So this idea was disproved by the works of Francesco Redi, the French guy Jablot, whose first name I forgot, Louis Pasteur and others. Redi did what? He took a piece of meat, put it in the tray. Took another piece of meat, put it in another tray. One tray was covered with cloth. Okay? So open piece of meat, of course, got a lot of maggots in it because flies were putting the eggs in it. The one that was covered with cloth had no maggots. So it was first blow. Okay? Jablot demonstrated that if you boil the hay extract, it never got the growth. And if you don't boil, it's got the bacterial growth. But the most convincing was the experiment of Pasteur. Actually took the flask, 2 flasks, with this shaped neck. So it prevented air from going in. Boiled both of them, then broke the neck of one. And it's got contaminated from the air, and there was growth and stuff like that. Second flask didn't get contaminated, remained sterile. It still does. It's in a Pasteur museum, more than 100 years. It's sterile. Pretty amazing. So, spontaneous generation was disproven. Now well, the next question is, where did life come from? But it's not a subject of science. Why? Why life generation is not technically a subject of science? Huh? Well, if we'll stick to materialism, then it's not. But why science cannot study the life generation? Why? How many? Yes. Huh? Well, no one was there when dinosaurs were roaming the earth, but we'll know a hell a lot about that. How many times did life appear? Huh? Once. According to all current concepts, once. Science doesn't work with single events. Science works with repeating events. That's the problem. It's actually not. Science doesn't care about origin of life. Who cares? Biology cares about evolution. Okay. So next thing to figure out was the cell, kind of self-explanatory. Schwann, Virchow, Schleiden, they all figured out cells, and that cells can be derived from other cells. And then came Chuck. You see, it says keystone of modern biology. It really is. Evolution is not just theory. You know that statement, right? Oh, it's just theory. Well, gravitation is just theory. You can test it, just jump out of the window and see how it works. Evolution is just theory, but it works. Not but, it just works. It is a law. It's something observable. Seriously. It's observable. So that's a keystone. Modern biology is based on evolution. Okay? Mendel was working pretty much at the same time as Darwin was doing. Mendel was studying genetics, and he proposed the first laws of genetics. Since then it was all changed, of course, modified, and other mechanisms were discovered, but Mendelian genetics is still important, as we know. Then other advance was the works of Semmelweis and Lister. Aseptic techniques. Okay? Have you heard about Lister? I bet you did. Not directly, but in the name of one thing that you can buy pretty much everywhere. Listerine, yes. It's not him who discovered it. It was kind of named in honor. But Semmelweis' story is more profound. He was the head of maternity ward in a hospital in Vienna. And a lot of mothers came down with puerperal fever. Ever heard about it? Essentially, it's a sepsis. Because when they give birth, blood is exposed. And if bacteria gets into the bloodstream, it's a sepsis and people die. No antibiotics. Okay? There's a good deal of deaths. He was puzzled and disturbed by this fact, and he noticed that mothers that were cared by midwives were better off than mothers that were cared by the medical residents, males, essentially, at that time. So, any ideas? What was the difference between midwives and medical residents? In terms of what midwives weren't doing something that medical residents were doing.
They were in contact with other patients.
Yes, and very special type of patients. Yes. Residents often came back to maternity wards directly from morgue. Well, they didn't have latex gloves at the time, so they just cut open the dead body and then go and deliver the baby. Kind of an insanitary, what do you think? So Semmelweis introduced the hand wash, chlorine hand wash, and the death just plummeted down. It was just amazing. His concept wasn't really welcomed by the society. Everybody thought he was a lunatic. And if everybody thinks you're a lunatic, you eventually become one. So he became very misanthropic and spent 2 last months of his life in the mental asylum, where he was placed by his wife and his best friend. Yes. It was mid-19th century. I wouldn't be able to tell you the exact years, but it was like 1840, 1850. So imagine before that, you do the surgery. It's like you can do it with a kitchen knife that you just use to carve turkey. It was pretty much the same level of sanitation. Nobody thought about it. Joseph Lister was first to propose sanitation techniques in the surgery room. He used phenol solutions, water solutions of phenol, carbolic acid, so-called carbolic acid, to spray on the wounds to prevent infections. It's rather barbaric technique because phenol is toxic and corrosive. And it worked, actually, and he was honored. He became what, a knight or something. He like had that many of regalia in terms of, he saved the life of one of the British kings. I don't remember which one. He had appendicitis and right before coronation, and Lister was a consultant during surgery. And people got sepsis after surgery like this. Okay. So that was a major breakthrough in terms of control of microbes. And then Pasteur. We know Pasteur for pasteurization of milk, right? Well, beer. In France, he was praised for saving French wine industry, because he was asked to help wine farmers or whatever. Wine went sour, like bad sour, okay? And he figured out that if you pasteurize it, certain temperature, it retains the quality, taste, but can stay longer in the bottle. He also discovered the vaccine, developed vaccine against rabies, and he first proposed the so-called germ theory of disease. So he was the first to say, look, we have a microbe, and microbe can cause the disease. Okay? Robert Koch established the guidelines how to link a microbe to the disease. We'll chat about it more. Just think about it for a second. Like, the fact that you have a certain microbe does not mean that this microbe causes the particular disease, right? So Koch showed how to establish it. Ivanovsky and Bergering discovered viruses. It was a huge breakthrough, because some infection like smallpox is not a bacterial disease. Meshnikov and Ehrlich in the beginning of 20th century made a major contribution to the science of immunology, how bodies protect themselves against infection. It's actually very interesting. They didn't like each other. Meshnikov, it was Russian-born scientist who worked in Paris, was the proponent of phagocytosis, cells eating other cells. And Ehrlich proposed that those were antibodies that protected us. They were crazy rivals. It turned out they're both mechanisms involved, so they both got Nobel Prize the same year. But until then, think about this. Get infection, and it's a pure luck whether you're going to survive it or not. No treatments. Well, okay, Ehrlich invented salvarsan that was used to treat syphilis. The drug was based on mercury, so it was toxic like shit. It was almost killing the patients. So it was awful. Okay? But then, in the 20s, Alexander Fleming discovered penicillin, and a company named Bayer, they invented sulfonamides, sulfa drugs. I think, yeah, both discoveries were made accidentally. Fleming definitely just noticed the fungus growing on the bacterial culture. There was clean area around that fungus. That makes sense? So that fungus produced some bactericidal compound. He managed to isolate it, and there you go. Got penicillin. Bayer, I don't know the exact story. Sort of an urban legend goes that the guy who was working in R&D at Bayer, his daughter was suffering from chronic wound infection. And he was desperate, so he used one of the dyes, actually. Like chemicals that dye things. It was some precursor of the dye, some compound that was like on the shelf to treat the wound. I don't know why he thought it's going to work, but it worked. And finally, in 1953, a bunch of guys contributed to the major discovery, major breakthrough in the mechanisms of heredity, gene expression, DNA structure, and stuff like that. And that actually, as you see, I pointed at it as keystone of modern biology. It changed our understanding of biological systems forever. I cannot say it's probably the biggest, well, I'm a biologist, I'm biased, but it's probably the biggest science advance of 20th century. It completely changed our understanding. Yes?
I got two questions.
Sure.
First is, the vaccination of cowpox is 9 days typical for someone to get an immunity to something like that, or?
Say it again.
So you said that he was inoculated 9 days before and then infected with cowpox.
Yes. Well, I wouldn't vouch for exactly 9 or 14 days, but yes, go ahead.
But is it typically a short period of time like that it takes to get resistance
to a certain infection? Right. Depends hugely. For some infections, it's typical. How many of you got the flu shot this year? Good. Me too. About 2 weeks for flu immunity. Huh?
Still got sick.
Any guesses on what percent? Huh?
Some people think they get the flu and it's actually just gastroenteritis.
Well, flu never causes intestinal disease.
Right.
Never.
Right.
Second, among respiratory infections, any guesses? What percentage does flu represent? Well, it's not qualitative, but how small? Well, come on, it's a wild guess. It's not an exam. You're not going to lose any points. Huh? 5%? Any other? In between about, according to different studies, 11 to 14. So everything else is a bunch of other diseases. Okay? So it protects, but not from everything. Okay? So other infections, rabies, the vaccinations, like 2 month, I think, schedule. Hepatitis B every 6 months. Thank you so much. 3 shots, yes. I missed my 3rd one. Never came back for it. So you see, it differs.
So second question is for the fermentation.
Yes.
I sort of assumed that wine having alcohol would kill off bacteria that would make it go bad. So what is the alcohol killing that the fermentation is?
Not at this concentration, no. It's just too low. 13%, yeah. It's just not enough. Okay? Now, probably there's another question that's being asked. What do we have to know? You have to know general idea of what these guys contributed to. I do not ask questions. Spell correctly the last name of the guy that did that, whatever. Okay, I'm not evil. But I may ask you, okay, which of the following scientists contributed the most to, I don't know, field of immunology? And believe me, I will not give you the option to choose between, say, Mechnikov, Ehrlich, and Jenner. They all contributed. So I don't want to, screw you over this. Okay? That will be pretty clear. So because other 3 guys will not have anything in common with immunology. Does that make sense? So have an idea what they've done. Okay? Now, what I found, any questions? I love when you ask questions, so please ask questions. So we're going to chat about a few things about chemistry. I found it convenient to kind of clear certain things before we go with mechanisms, cellular mechanisms. Remind you, okay, about basic chemistry of life. Water is everywhere. Well, like what, 60-70% water? Bacteria is like 99, something like that. They have a lot of water in them. And the fact that we look like this, walk like this, talk like this, and the life around us is like this, it's because of the special water properties. So remember that water molecule, one oxygen, two hydrogens, is partial charges. Oxygen is partially negative, hydrogen is partially positive, which makes it a polar molecule. So those negative charges, or negative charges, will interact with positive charges, which means if you throw in iron, positively charged sodium, cation, it will get hydrated, okay? If you throw in negatively charged ion, it will also get hydrated. But what is also important is that those water molecules will interact with themselves. They will establish hydrogen bonds. Okay? Hydrogen bonds are formed between the partially positively charged hydrogen and negatively charged something else. Seriously, something else. Like in ammonia, hydrogen is partially charged ammonia. Hydrogen is partially charged, partially positive. Nitrogen is partially negative. Can ammonia form hydrogen bonds? Sure. This hydrogens will, well, partially, delta, will interact with nitrogen and stuff like that. Okay? So hydrogen bonds are responsible for a lot of things in a cell. For holding two strands of DNA together. For actually hydrating those DNA strands. For shaping the proteins. It's all, well, not all, but a lot of it is hydrogen bonds. Okay? Now another thing that we have to remember is pH. pH scale. What does it show you? Excellent. I haven't heard this word in a while. We're going to refer to it as the proton. Okay? It's hydronium, yes. So, concentration of that ion. Okay? Now, I don't want to go into the math of pH. It's a negative logarithm of the hydrogen ion concentration. But anyway, keep in mind that this is your pH scale. No, I will not ask you the urine pH or the coffee pH. I don't really care. What you have to know, though, is pH lower than 7 is good. Higher than 7 is basic or alkaline. 7 is neutral. Water, if you leave it on the table, what pH is it going to have? Like distilled water. 6-ish. Why it's lower than 7? Excellent. Carbon dioxide from the air. Does that make sense? What's the pH of pH of life? Huh? It's very, very, it's much, well, life, okay, human life. Higher. 7.46, well, 6 is a little higher. So pretty much we talk about very narrow 7.4. We maintain very, very precise pH in the blood and extracellular fluid. Microbes, they can live everywhere. There's one guy that lives here, so-called thermoplasma. Huh? Say again? Oh, Helicobacter pylori. H. pylori survives at 3.2. Thermoplasma beats it. It lives in the acid runoff from copper mines. It lives in acid. It's pretty amazing. There are some that Nitrobacter lives here, pretty much. So they can live virtually everywhere. Now, molecules of life. Sorry. Carbs. Carbon, hydrogen, oxygen. Arranged in a very specific way. So you have monosaccharides, okay, that they can combine into the disaccharides, right? And some disaccharides are important food sugars. That's a sugar right here, sucrose, okay? That's maltose, which can be found in any malt-containing beverages, for example. Lactose is the milk sugar. And then, sorry, monosaccharides can form long branched or unbranched chains. Amylose, the linear polysaccharide. Starch that you're going to find in the, what's the name? Plants, right. Glycogen that you're going to find in animals, okay? So, but this is the exemplary molecule of monosaccharide, okay? You can see that oxygen, boy, the cyclic form of that molecule, okay? So, a lot of hydroxide groups, and they form long chains via the dehydration reaction. So hydrogen and hydroxyl groups go away in the form of water, and you have that ether bond right here. Okay. Now understand pretty much the basic idea that I want you to remember is that we have monosaccharides that can be combined in the longer chains, right? They can be hydrolyzed back in the monosaccharides by water and usually enzymes. And their major utilization is energy, of course. But they're also part of cell structures. They sometimes are attached to lipids, to proteins, okay? And they are part of DNA and RNA. That make sense? Any questions on the carbohydrates? Yes?
The question is, are you going to make us have to draw any of these?
Oh, come on. It's not a chemistry class. Look, this is my point. I try to, if there's memorization, it should be meaningful memorization. There was something in a, yeah, like, you know, all 20 amino acids. Can I make you memorizing all of them? Sure I can. I'll tell you it's going to be on the exam. And you will spend hours memorizing the structure of 21 amino acids. And then I'm going to ask you and you're going to answer. What's going to happen to that knowledge once you step out of the class? Yep, it's going to be gone. Because you're not going to use it. Unless you use it, you lose it. Right? So that's, unless you're going to work as in the chemistry of carbohydrates, or, sorry, you're going to work in the food industry, okay? So you're going to actually refer to those structures. Come on, that's the point. Lipids. We refer to them as fat sometimes. There are two major types, I'm sorry. Triglycerides and phospholipids. So triglycerides are illustrated here, okay? It's a hell of a picture. So you have glycerol, which is the alcohol, essentially, with 3 hydroxyl groups that forms ether bonds with 3 molecules of free fatty acids. Fatty acids can be completely different, they can be same, it's all different, okay? Depending on what fatty acids you use, the fat, the triglyceride, is going to have different physical state. Any triglycerides in your kitchen? Shortening, oil, butter, those all triglycerides. Depending on the chemical composition, they can be solid, like shortening or butter. They can be liquid, like say olive oil. Okay. They are essentially the best energy storage. Store a lot of energy, but to take it out, you really have to work a little bit. They're not the primary energy source, but very valuable. Can be broken down. So this reaction can go back if you have proper enzymes, or if microbes have proper enzymes. That makes sense? So it's both ways. Phospholipids, composed of the glycerol here, 2 free fatty acids, and a phosphate group. They are essentially elements of all membranes in all living things. We talk about cellular membrane in bacteria, phospholipids. Archaea, phospholipids. Eukaryotes, phospholipids. Not only, but that's the staple, okay? They not energy source, no, but still important. Sterols. Well, cholesterol is the part of the eukaryotic cellular membrane. It regulates its rigidity. Membrane can be more or less fluid, more or less rigid. That will influence the cell shape, the transport across the membrane, all those things, okay? Analogous structures, similar. You can see there are some similarities between sterols and hopanoids. Hopanoids regulate the fluidity of the bacterial membranes. Does that make sense? So that's your lipids. That's so far what we really have to know. Triglycerides, phospholipids, sterols, and hopanoids. So again, know the basic function. Membrane rigidity here, fluidity, whatever. Membrane structure, energy. Any questions? Now, going back to the question about the structure. Will I ask you to draw? No. But I will presume that you know the detail like this. So if I ask you, I show you a picture. Of the molecule, and I'm asked, what is that? So you have to recognize the structure. So this is triglyceride, okay? This is phospholipid. Does that make sense? So you have to recognize the structure, but I will not ask you to draw this. Clear? We good on that? And the proteins. I'm going to ask again. I have really bad memory. Somebody took 1200 or 1510, right? How many amino acids we have? Well, don't look at the PowerPoint. 20. It was 20. Well, in fact, we have 21. We have 21 amino acid. 20. I was taught when I was in college, we learned 20. Turns out there's 21st amino acid that was discovered like early 90s. It's called selenocysteine. So it looks like this. Instead of sulfur, there is selenium atom. It's actually very important. Present in all life forms. So those amino acids form polymers. They form peptide bond between hydroxyl group of one amino acid and amino group, hydroxyl group of one amino acid and amino group of another. They interact, it's again dehydration, form peptide bond. You see there's a common motif here. every time in life you form a polymer, you remove water. Whether you form a triglyceride, you form a polysaccharide, or form a protein, you remove water. So in order to break them down, you have to add water. Does that make sense? Proteins fold. I don't even know how to give an example of the real life thing. I have none. Essentially, you put amino acids in line, and based on the structure, so-called side chain, they will start to interact with each other, and they will start folding. So the sequence of amino acids, the way they are lined up, is called the primary structure. Then they fold, and they can form two major types of secondary structure. It's alpha helix and beta sheet. Okay? And then those alpha helices and beta sheet will start folding, forming a globule. Frequently, it's the last step. That's tertiary structure is the last. Sometimes proteins consist of several globules, not chemically bound to each other. Like Lego pieces, they're not glued to each other. You can put them apart. Do you know of a human protein that consists of 4 separate protein globules? I can guarantee you've heard about it. Think blood. Hemoglobin. There you go. 4 separate globules. But it's considered to be one protein. Okay? Now you have to have an idea what each level of structure means. Primary is the sequence, secondary alpha helix is beta sheath, tertiary is globule, quaternary is several globules together. Why pay so much attention to that no doubt interesting factoid? How many of you, well, it's kind of a personal question, okay, do you cook eggs for breakfast? Not for breakfast, something else. Uncooked egg, how does it look? Huh? Runny. Okay, runny. It's like liquid, right? You throw it on the skillet, what happens to it? Solidifies. Why? That's one thing. Something else happens. If you will just dry the egg, it's not going to look like you fried it. Huh? They denature. Well, they stick together, but they stick together because they change the shape. Okay? You denature them. You break down the structures. Does that make sense? So that's protein denaturation. It's a very important concept when we talk about life, because we function as protein bodies. Look at us. Well, this is protein, skin, a lot of proteins. Everywhere there are proteins. And if they denatured, we're screwed. Actually, one of the examples that a lecturer in biochemistry provided us when I was in college, I bet females know more about it. The curly hair, like you go to the hairdresser and they make your hair curl, I don't know, the perm. Okay, you do perm. How does it work? What do they do? I've never done, well, you can see. Chemicals. Very good. What happens with perm with time? It kind of, unwinds. So what the hell happens when you do perm? Well, not this, obviously. Huh? Say again? Absolutely. Absolutely. They denature keratin. And they denature collagen. They denature proteins. And then with denatured protein, you can change the structure of the hair. Okay? Same goes when somebody straightens the hair. You denature protein first, straighten it up or curl it up, and then with time it assumes the shape it's supposed to be in. So that's why perm goes away. Does that make sense? So that's important. Denaturation is the fundamental feature of the proteins. And nucleic acids. Well, we didn't talk about structure. What are the functions of the proteins? What do they do? They can. Energy. Something else. Huh? Okay, building. So structural function, right? Something else. Mentioned that word a few times before. Chemical reactions in the cell cannot run without enzymes. Okay, enzymes. Those reactions are freakishly fast, and they fast because of the enzymes, chemical catalysts. Okay, so proteins represent the majority of enzymes in the cell, right? So nucleic acids, DNA, RNA. Those are, yes.
This is a little bit back, but when you were talking about polymers, what specifically were you saying? What would you consider a polymer?
Definitely glycogen, starch, amylose.
Okay.
Now, good question. Where does it become a polymer? So like,
Plastic.
well, plastic is the polymer too. Polymer is any molecule that consists of technically more than one building block. Building blocks can be the same, like polyethylene. It's going to be homopolymer. Building blocks can be different, like protein, amino acids are different. It's going to be heteropolymer. Now, if you have like 2 amino acids, we call it, it's a dimer. 3, it's a trimer. When does it become a polymer? It's the same question as when a few becomes many. Does that make sense? So like if you have 100, it's a polymer. If you have 10, it's up to you. You can call it a decamer or you can call it a polymer. But essentially you have many, many building blocks, like a train. Okay? Last thing that we're going to chat about is the nucleic acids. We're going to talk more about structure, but what I want to remind you before we can move forward, of 2 types, major types, DNA and RNA. They all consist of just 4 nucleotides. Just 4. They're slightly different between DNA and RNA. DNA is AGTC, and RNA there's no T. Don't focus on that yet. We'll get to this eventually. Important thing is that those nucleotides can pair complementary. A always pairs with T or U. G always pairs with C. That's the correct pairing. Right? So it's a fundamental feature of life. Now DNA, what's the function? Codes for proteins, stores genetic information. So technically speaking, like we have a genome of smallpox. We know the whole genome of the smallpox virus. But technically and it's gone in the whole world. We don't have smallpox anymore. No virus. We can recreate the virus. It's technically possible. Not needed, but possible. So by copying the strands, I don't have an illustration of strand copying, but by copying strands, we can reproduce the information, give that information from one cell to another, from one virus particle to another. Does that make sense? That's a fundamental feature. And assuming DNA polymerase doesn't make any mistakes, the new cell if it's not sexual reproduction, and we're not going to talk about sexual reproduction at all, we're going to talk about pretty much basic binary fission, okay? If it's like regular division of cells, new cells have the same genetic makeup as the old ones, okay? Now, that DNA has to be, it encodes for proteins. Proteins are the result of expression of genetic information. That expression occurs by the messenger RNA. We'll talk more why there is an intermediate step between DNA and RNA, and protein, sorry. Why there is RNA. What is the reason? Why it's convenient? Why not directly? Okay? Huh? Right. It goes from nucleus to the cytoplasm if it's the eukaryote, and in prokaryotes it's much simpler and faster. But pretty much the idea is that every time you have to make a protein, you can only express one gene instead of dealing with the whole, manipulating the whole genome, which is big. I mean, size-wise big, like physical size. You can stretch out DNA several meters. So it's like physically big. Okay? So, we're not going to go next, but just remind you, we have to think throughout the course, when we talk about anything from control of microbes to antibiotic resistance to virulence factors, we have to think about DNA being transcribed into RNA and RNA being translated into proteins, that flow of information in a cell. And somehow we have to include evolution there. Okay? So those are, I don't know how to emphasize it more. There's a keystone things. They're going to go like a red line through the whole course. Evolution, gene expression. Okay? Well, I will see you on Thursday.